cases that you had, that I thought I should really work harder on it, but even the second time I really didn't have it. But at that time it was not possible to talk to Russians because they were everything was closed still. And when finally I had Russian colleagues, they didn't want to talk to me in Russian because it was painful for them. So maybe in the future I have the chance to pick it up again. And
you start to talk in Russian.
I understand a few words, and that's everything that is left. Okay, so yeah, my group is now in Göttingen. We were until 2 years ago in Munich, and I wanted to give you just a short overview. So these are all the PhD students and postdocs in the group. I wanted just to tell you what they are working on, so that you have maybe you also want to discuss other things. I'm really happy to be here at such a PhD school, also because it's nice to get to know many people. And so if you want to discuss anything afterwards, please feel free to talk to me. So Marcus is working on remote homology detection. That's what I'm going to talk about today also. And on an algorithm for global domain decomposition. So we want to find out where the structural domains in the sequences are. And this is a heavily statistic-based algorithm. So a lot of what we do is really very much statistics. But also some algorithms, and that's also the second part of the talk that I'm going to present. So Martin is a student who's also here, and we'll talk about this very algorithmic aspect of our work. So then we work on Then we work on statistical methods for contact prediction. So we are looking at co-evolution of columns or positions in a protein that we can analyze in a multiple sequence alignment. And this has been started by Martin Weigt and Marx and Sender something like 4 years ago or so. And is the first method that is really reliably able to predict de novo the structure of proteins, the fold of proteins. It's not very high resolution, but it's quite reliable. If you have many sequences, that's the big if, and that's why we are working on it, because we want to bring the number of sequences that you need down from now about 1,000 to, hopefully something like 300 or so. But we're still working on it, we're not yet successful. We're using a lot of Bayesian statistics there. And Martin, I'm going to talk about this project. Clovis has just recently started and works on MMseqs and on applications to viral metagenomics in a project that we have recently joined, a European project called VirusX. And Lukas works on the detection of activation motifs for transcriptional activation. So these are short linear motifs that bind to targets like, for example, TF2D, a cofactor for transcriptional activation, or mediator, or other targets that help transcriptional initiation rates to go up. And we have a collaboration with Steve Hahn at the Fred Hutchinson Cancer Research Institute to, an NGS experiment to find out, to generate random sequences, put them at the end of a DNA binding domain and see how strongly the transcription is activated. And we're analyzing at the moment these data to find out which motifs activate, what they have in common. So then we work on Bayesian motif discovery in DNA and RNA sequences, and I'm going to talk about that tomorrow. And on systems medicine using Bayesian statistics, so we're looking at GWAS data and trying to integrate it with mainly EQTL data and functional genomics data from ENCODE. And the goal is to find those genes and gene sets whose up or down regulation is predictive of disease risk. So those are the ones, the possible targets of pharmacological treatment, right? If you know that certain genes are upregulated in high-risk patients, you probably want to investigate whether bringing the levels of those genes down or lowering their activity can somehow prevent cancer, prevent disease. And this is a general method, but we have partners, medical partners that work on atherosclerosis, so that's our model system. And we are developing methods on single-cell transcriptomics, and this is again very heavily statistics-based, and also on core promoters. So core promoters and also thermodynamic models of DNA binding. And we are always looking for good students, so if any of you is interested in a postdoc or so, please contact me. We're also collaborating a lot with experimentalists. I haven't mentioned this here, but we have, for example, at the moment we're analyzing a lot of par-clip data, so similar to ChIP-seq, but for the transcriptome. So we're immune precipitating factors that bind to the RNA, then we're looking at the RNA sequences that come with it, and developing methods to distinguish the signal from the noise from the background. So the topic of today's talk is sequence searches, and I think homology is the reason for why sequence searches are so extremely powerful in protein bioinformatics is because structures and functions may be conserved over billions of years, and billions of years really means that you often find a template for a protein sequence that you want to predict the structure for that is from a different domain of life. So you often find some protein in eukaryotes. You don't find any template from the PDB that you could use, but you find something in bacteria or in archaea. So this is really not rare, and one example is given here. So this is ubiquitin from yeast and from humans. They are almost identical in structure, although the sequence is relatively diverged. But even SUMO, which is a functional paralog, you could say, of ubiquitin, is still in structure has a RMSD of, I don't know, 3 or so angstroms. So it's still very, very similar, even though it's doesn't even have the same function, and is both present in yeast and in humans. Yeah. So the first reason is that this enormous conservation, and I will say a little bit about why the conservation is so good. And the second reason is that if we have two homologous sequences, we often can, you could say, almost prove that they are homologous. It's not a proof; it's a so-called hypothesis test. But when this hypothesis test gives us strong evidence, then we are almost never wrong. We have to be careful about the statistics, but this gives you very reliable predictions when you have homology. And whenever you have homology, and you still can detect it in the sequence, then you usually have a very similar fold, and often, not always, but often a similar function. Okay, so this is the power of homology and sequence searches. So this role of sequence searches is, of course, to find those similar sequences and to use them for predicting the function of your protein of interest. So just to give you a few examples, a few years ago, in 2011, two papers came out back to back in Science, and they had both used HHPRET to find the function of a protein that was known to be involved as a general transcription factor in POL1, and they found it is, so this is called TFB in humans and RRN7 in yeast, and they found that it was full-length alignable in HHPRET with a protein that is very, very well studied from POL2 called 2F2B. So the name B here is just a coincidence, and so they found that it's actually a very close functional paralog of 2F2B, but for the POL1 system. And then they did a few biochemical studies showing that it does more or less the same thing. So this was only one sequence search. They took the TF2B and searched against, searched against the same genome. So they took TF2B from yeast, for example, searched against the yeast genome, and found this more or less full-length alignment. So basically a matter of a quarter of an hour or so. Another example is the elucidation of the structure of the type 6 secretion unit. So this was mainly done by Piotr Gleiman. I guess he's Russian, I'm not completely sure. At least his name is Piotr. And he was still in Lausanne, and he was moved to Texas. And what he found is many homologies between the type 6 secretion system in bacteria that was discovered only, I don't know, 6-7 years ago, and parts of the phage T4 needle and spike system. And so by finding these homologies, he was guided to solve the structures of these pieces one by one and assembling them and so on. And yeah, I still communicate with him quite often because he's still very grateful, even though we haven't done anything but just offering the server. So I think these are maybe the more prominent examples, but it shows you that even very deep homology is often a very powerful method to predict functions of proteins. Of course, then you always have to verify whether this prediction makes sense, but as I said, often it does. And a nice example that goes down even deeper than the common ancestor of the 3 domains of life is shown here. So here we found, and we found actually many similar examples. These are 2 protein folds. One is the AAA plus ATPase fold. It's a big family of mechanical motors. So upon ATP binding, this C-terminal domain moves. And it's, for example, the basic working unit in helicases and I guess also topoisomerase. Many motors that work in the cell. And this is the histone dimer fold. And of course, both of them are very central to life. This everywhere and this in eukaryotes. And what we found when we looked at, it's quite a long time ago, in 2003, we looked at homologies in the universe of known proteins where the structure was known. We found this homology and we thought this is a mistake. But then luckily I saw that, so this is one chain here. Luckily I saw that this match to one chain was actually, it was to this guy here. So one chain of the histone dimer. And I found that actually this, of course, is not structurally linear to this, but this upper part is. And so what happened is that at some point the C-terminal domain must have lost its kink here and straightened out. And by dimerizing, it regained exactly the interactions that it had in the original fold. Okay, so if you align them structurally, they are very similar. And so you have both, and in this case, both sequence and structural evidence that this is a homologous piece of protein. Okay, so we've built a server that was already published in 2006 that is very widely used, and the way it works is it takes the protein sequence, it first builds a multiple sequence alignment, and these can be closer homologs. They don't need to be present in the PDB, they don't even need to be annotated at all, but having this profile, or this multiple sequence alignment, you compare it with other multiple sequence alignments, and this is what gives you the power, and I will show you in a moment why this is so. So you don't compare sequences to get this sensitivity, you compare multiple sequence alignments with each other. And the way you compare them is you compress the information into a so-called sequence profile. So we have the 20 probabilities for each amino acid in each column, and then you do some kind of comparison between those columns, and then you come up with hits, and these hits can then be so matches, for example, in the PDB, and when you have proteins with this sort of structure, you can use them as templates for homology modeling. So we derive probabilistic restraints from the alignment, and using those probabilistic restraints we can generate a model. How this homology modeling works is, do you want me to explain this? Please lift your hand if you want to have the idea how homology modeling works. So I guess if there's only 5, I'll skip it. I guess most of you will probably know. And we've also worked on this part to improve this. So we are using the modeler package where we've improved the probabilistic restraints so that modeler can work better with multiple restraints. The server, as I said, is quite widely used, also because it's very simple. So you paste your sequence, you choose a database. You can also choose many genomes. Now this is an old slide. Now there's a window next to it where you can choose genomes of model organisms, and then you get a results page that looks like this. So here you have a summary of the most important hits, and then come all the alignments. For each of those lines there's one alignment, and the upper part always shows you the query sequence, or the query profile actually, and the lower part shows you the hit in the database. And there's a lot of information that you can access through a help button that's on the upper right corner, so it's, I think, quite well explained, and I don't need to go into any detail here. There's also a histogram view, so by clicking on this little symbol here, you go back to the alignment view, and by clicking on the alignment view at the symbol, you go back to the histogram view that shows you which patterns have actually been picked. Because as I said, it's not the sequences that are compared, it's the patterns, the profiles. And for example, this succession of hydrophobic and charged amino acids in this case is quite a strong pattern that is picked up by the algorithm. As an example, that we use for one of the publications, this is a protein that was a function of a domain of unknown function in PFAM, and we looked at whether we find templates in the PDB. They had a predicted internal transmembrane helix. Otherwise, there was nothing known. So we used our homology search and predicted that it actually contains, apart from the helix we also found, but we found two domains. You see a green one inserted into a blue one, and the blue one is a protein kinase domain, and the green one is an EF hand. And probably also because of this insertion, which is rather unusual, it's the first protein where this insertion is known, it was not picked up by other less sensitive protein search algorithms. And so just from such prediction, now we can of course use it for homology modeling. These two templates can be used, that were found, can be used to build the model structure. This is the result for the modeling. And just from half an hour of work, we can predict that it's a protein kinase that probably, because we also think that it's still functional because it has all the residues that are needed for a functional protein kinase, that is somehow regulated by calcium 2+ concentration. And this makes sense because it's bound to the ER membrane probably, and this would be a probable or likely place to regulate a kinase using calcium 2+ concentrations. And there are two further homologs in humans that also share the functional residues. So probably this is a family of functional kinases. Yeah, just to make a little advertisement, this. So the last time I did this analysis was CASP-10. This is already, I think, almost 4 years ago. We're participating every time in CASP. It gets a bit more difficult for us every time because now everybody uses our software. So this is the mean response time of the servers in CASP-10. This is the average model score that the server submitted. So the servers get sequences, but they're not told anything else. And then the servers have to send back their predictions and their model structure, their 3D structure. And the CASP organizers make sure that the structures are not yet solved when they are sent out, so nobody can cheat. And by knowing the experimentalists, you don't have any advantage. And then, so each dot is a server, and 4 years ago at least, we were one of the best servers, or all three were among the best, but much faster because we are not doing a lot of post, let's say, we build a model, but we don't refine it anymore. We are building the model with a modeler in our improvements, but then we let it at that point, and most servers take a lot of time trying to go from there, building hundreds and thousands of models, clustering them, and so on, trying to find the best clusters and so on. And most of the tools at that time, most of the servers used our software, and that's also why it gets a bit harder, and we're not so super ambitious anymore in participating, although we have the server still running this year. Yeah, and I think because the server has a lot of functionality that is nice and flexible, so you can change at any point of the path between your sequence and the model, you can make changes. You get suggested, for example, a good alignment between your query and your template, but you can also change it by hand. And I think because of this flexibility and because it's fast and quite user-friendly, it's cited quite a lot. So let's see where we have enough time. So when did we start here? How much time do we have? Okay. So maybe I go quickly through this because I think most of you come from a biological background, right? So you have a good feeling for proteins, I guess, also, right? So maybe I'll just give you my whirlwind tour through why I think that proteins are so good candidates for homology search. So this is a ferredoxine protein, very simple fold, colored by where each amino acid in the backbone is colored by its physical chemical properties, so into aliphatic, aromatic, small, polar, negative, and positive. And when you look at the space filling representation of this, it looks fairly dense. I think in proteins 75% or so is filled, and the rest is water. But when you take away everything but the aliphatic, aromatic, and small ones, you see that there's still quite a dense core. And I think that and this makes sense, of course. We have mostly hydrophobic interactions in the core of the protein. And this core, I think, is what makes the folding logic of a protein and what makes the pattern so stable. Because if you look at typical alignments that go at least hundreds of millions of years in this example, you see these patterns conserved usually through these hundreds of millions of years. Not completely, but mostly, let's say. And I think it's these patterns that make it so nice for us to look back billions of years, because there are not so many ways to build a protein in a stable way. And you have to imagine all these protein folds that are around in all these different bacteria and other animals. The folds are competing with each other as well. It's not only the organisms. So if you have a mutation in one of those, let's say, bacteria, and it gives you a protein that is not as stable anymore. For example, if it's an enzyme, it just needs to be stable and perform its function optimally to have the right steric orientation for the functional site. If it's not as stable anymore, it won't perform its function as well anymore, and you will have a slight disadvantage of the individuals that carry this protein. And because of the population numbers in bacteria, this will get outcompeted. So if the proteins are also underlie high pressure, and so that's the first part. And the second part is there are not so many stable ways to make a protein stable, because you have to find a solution to this, what I would call 3D jigsaw puzzle. It's like, not a 2D but a 3D puzzle, and there are not so many ways to make this stable. And we know that because when we generate random peptide sequences, they almost never fold into something stable. If you put in enough hydrophobics, they will fold into something called a molding globule. That means through the surface tension of water, it wants to minimize its tension, it will just push the protein together. And when it's hydrophobic enough, this will work quite well. But it's nothing stable. It won't give you something that is able to perform a biochemical function very well. So I made this argument now for enzymes, but the same is true for proteins that undergo allostery, only it's more complicated. Then the selection is even on different conformational ensembles that the protein has to undergo. So in a way, you could imagine the folds of proteins as being the tips of islands, and only some small pieces come out of the water. These are the ones that are really optimized well over hundreds of millions of years. And if you want to change the fold, you have to somehow swim through water. So you have to go through a lot of space that is not optimal anymore until you reach a different fold that is stable again. And so evolution doesn't want to go through this water, of course. And you always have to, so this time that you go through the water, you have to make sure that the protein doesn't degrade completely, is completely useless, and also so usually that it performs some function so that it's selected out. And that is very difficult, and that's why we have relatively few changes or let's say inventions de novo of new folds. Usually those folds that get invented are repeat folds where the unit that you have to invent is really short, and then just gets amplified and gets something folds into something stable. But more complex, non-repetitive folds usually don't, at least by our conviction, don't occur de novo. And of course now it's clear to see that if you find something homologous to fold X, then it will usually be again something that has fold X. Okay, so I'll just skip over that. And I would want to now tell you that why we are using alignments to compare with each other in our sequences. So one thing that is maybe interesting to give as an argument is, why can we be so sure sometimes that two sequences are homologous? As I mentioned, we are using a hypothesis test. And the hypothesis test is, I would say, the most important concept of classical statistics. It means so let's say you got a score for these two sequences, for example. Let's say you got a score for these two profiles or sequences, doesn't matter. And you get a certain similarity score. This is what I call S here. And you know the distribution of your sequence with many other sequences that you don't, for which you think they're mostly not homologous. So you've got a distribution over scores for mostly non-homologous proteins, and your score is much higher than most of them. So this area here to the right is called p-value. That's the probability that comparing to something non-homologous, not related, you'll get a higher score. So if this p-value is small enough, then it gets less and less likely that your score is due to really a chance event, that it really follows the solution. But it gets more and more probable that it's not due to chance, but it's due to some common ancestry. Okay? So of course we have to do a correction for, if we do many comparisons, we still can end up very much on the right. So we have this kind of something called bound-Rome connection, or another interpretation is the factor that stands between the e-value and the p-value. So for example, if you compare with 100 million sequences in the database, then of course it's fairly likely that you will get a p-value that is, well, 100 million is 10 to the 8. It's fairly likely that you will get a p-value of 10 to the minus 8 or a bit bigger. So you have to correct for this 10 to the 8, and that's that gives you the e-value. But when the e-value is, let's say, 10 to the minus 5 or so, and your statistics is more or less okay for your, this distribution was correctly taken, then you can be fairly confident that gives you a homologous pair, and that you can use this protein to predict your structure or your function. Okay, so I skipped that. That's just an exercise for calculating the p-value in practice. So structure and function of the protein domains are often conserved over billions of years. That's one ingredient. Sequences are diverged beyond recognition at those timescales. But we can develop tools to uncover the homologous relationships by comparing multiple sequence alignments of closer homologs. So how do we do that? And this is the topic of the next small section. So first I want to just motivate that most of what we do in protein bioinformatics is not done with on the single sequence level. But almost always we first search for homologs, build a multiple sequence alignment, and then we do our predictions. For example, secondary structure prediction, membrane analysis, everything you can think of. Because most of these things that we want to predict for a single sequence are conserved among closer homologs. So it makes sense to take them into the game and do a prediction for all of them. So all of these things are done with alignments. And so sequence alignment is in principle a very simple thing. You have a certain similarity for each pair of amino acids, for example for the V and the I, and then if you find the right way to put them on top of each other, you can just calculate the score by summing up those pairwise similarities, so this is shown here, and then subtracting penalties for the gaps. And the way you can do this, so there's an algorithm called Smith-Waterman, or it's also sometimes called dynamic programming, and it's a more general algorithm that can calculate the alignment that gives you the maximum score. Now please hold up your hand if you know what the Smith-Waterman algorithm is and if you know how it works. So this is quite few. Then maybe I should explain, because I would make it, I think the daring point that the dynamic programming algorithm is probably the simplest, the best combination of a simple algorithm with a powerful algorithm. It's not only used for sequence alignment, it's used for many things in bioinformatics, also for example for structural alignment. So let's go quickly through this algorithm. So it's one of these iterative algorithms where you start. So maybe I should first introduce this dynamic programming matrix. So you have the sequence X on the left and the sequence Y on the right, and each cell gives you the score of this one amino acid aligned to the other. And for example, the score of R and K, arginine and lysine, is relatively good. And a mismatch, for example, of a hydrophobic isoleucine and lysine gives you a bad score. Now you want to find the path through this matrix that gives you the maximum score. And of course, the path is just the way, it's just alignment, writing the homologous amino acids on top of each other. So how do you find this path? And the answer is by the Smith-Waterman algorithm, and it works by starting at the top left cell and working yourself down, always using the same simple calculation to fill in the next cell. And so suppose we've already filled in all those gray cells and we are trying to fill in the next one here. So for each of those cells, what we've calculated is the score of the best score of the alignment that ends at that cell. So in the beginning it's very simple. If the score here would be a virtual zero, and the best score here would be the match score between x1 and y1. Except if it's smaller than 0, then you would just say it's 0 because there's no alignment there. So suppose you've filled out all these scores and in this matrix, and you come to this place here. Then the score of this cell is the maximum of 4 values. 0 means you are not able to give an alignment that has it's basically a length 0 alignment. It doesn't make sense to have a negative score because the alignment can always stop. It is a local alignment, so it can start and stop at any position. So 0 is the lowest score you can get. But the second possibility is that you had that the best score that you get is by coming through this cell here. So then the score for this cell would be, of course, the score for this cell plus the match score for xi and yj. So that is the best score for I-1 to j-1. This is this cell plus the match score for xi and yj. But you might also have a gap. So this is this first possibility here, that xi minus 1, y-1 were aligned and xi and yj. But the second possibility is that you came through this field here, and that would mean that that what you actually align at the last position is that you actually have a gap between, so that xi is not aligned to anything, and that you just take the best score that you had up to here, and you subtract a penalty because now xi is not aligned to anything in the y sequence. And so that is this possibility. So vi minus 1j is the best score that you had until here, and now you have to subtract a gap penalty because you align xj to nothing. And the same in the other way around is also for this holds for the green arrow, and then you have 4 possibilities. You take the maximum, and that tells you how to fill this, and then you go up to the very end. And when you do that, so here is an example where we just have a match penalty of +1 and a mismatch of -1 and a gap penalty of -1. And so this is what you get when you fill out this matrix, and in this case it's simply nucleotide. And you come up with, then you look for the maximum score in this matrix, and we know now this score tells us that there must be an alignment that ends at this matrix, and that has a score of 5. Okay, so that's the sum of the match, the mismatch, and the gap penalty score. And then you just work yourself back and see, well, from which cell did I come to get this 5? Well, I came from here, and so on. From which cell did I come to get this 4? I came from the 3, and so on. And then this is called backtracing. Working yourself back from the cell with the maximum score, you find the alignment that is guaranteed to give you this optimum score. Okay? So it's actually very simple. I mean, maybe it sounds complicated if you don't have enough time to think it over, but it is actually one of the simplest algorithms that gives you highly non-trivial results, because it's at the basis, at the core of all sequence searching algorithms. Of course, we don't use match +1 and -1 in practice, but we use substitution matrices. So there's just the lower part shown because it's symmetric, and this just, these just give us statistics that is then calculated into a score. So the statistics is how often you see 2 amino acids aligned with each other in many training lines, divided by the probability, the independent probabilities of those 2 amino acids. And if you take the logarithm of those 2 probabilities, you get something that is positive if they like to be aligned to each other, and negative if they tend not to be aligned to each other. Okay, so this gives you a natural score that we then use as a quantitative measure of similarity of 2 amino acids. So now we can also go to more interesting cases, because I told you, let's skip this, I told you that we are interested to align amino acid alignments with each other. So how do we do that? Well, first, to motivate this, when you look at these 2 sequences, they get an e-value of 0.2. When you first take the green sequence and you do one search through the database, you get this alignment, and when you now recalculate the score for this alignment with this sequence, and I'll tell you how to do that, you get an e-value of 10 to the minus 17 all of a sudden. This e-value is not really significant enough to believe the hit, but this certainly is. So just by adding a few sequences, you get an enormous amount of significance in addition. So information is power, and this is actually true everywhere in bioinformatics. And I think it's a very good exercise, or a very good habit, I would almost call it. If you're considering a problem you're working on, for example, you want to predict something, you want to make, want to derive information from a data set, to think about where the information is hidden in the data set. Because often people improve dramatically on previous work by realizing that there's some information that hasn't been used before. It could be other data sets that could be integrated into the data that you have, but it could be just because some aspect hasn't been really hasn't been put into the model that is used for the analysis. So information is power. That is something that has guided me at least for most of my work in biomechanics. So now I wanted to tell you that this algorithm is actually not only able to align sequences, but the same algorithm can be used to align profiles. So a profile with a sequence or a profile with a profile. And I told you a profile is just a matrix where the 20 amino acid frequencies in each position of the alignment are shown. So when you do a profile alignment, you take the profile score. You just substitute the profile score for the amino acid score. So here now we have a profile that we want to align to the amino acid. And instead of taking this probability of seeing the 2 amino acids in a large alignment of a large collection of training alignments, we now take the frequency of this amino acid in this column J of the alignment. So the alignment that is underlying this profile. Right? It's again a so-called log odds score, because these are odds, meaning the odds ratio of this probability and this probability. This is the model probability, this is the null model probability. So this is the probability of finding xi at this position J of the multiple sequence alignment, and this is just the probability of finding the same amino acid anywhere in the database. The ratio of this again tells you how much this amino acid wants to be at that position. And then the rest of the algorithm is the same. And it's really amazing what such a small change in the scoring can make in terms of sensitivity improvement. And then you can do the same trick, but now comparing one profile, a query profile with a database profile, for example. You just need to put another score here. And I don't want to discuss what kinds of scores you can put. Our favorite is this one here, but it doesn't matter. I think you understand the principle, right? You need to have some comparison metric between one column of one sequence alignment and another column of another one, and try to put the most similar ones on top of each other. That's what the dynamic programming will give you. So this is again a profile. You see all the scores are pre-calculated, and they're basically what you would get. For example, the y here minus 3.2 is what is the score you would get if you wanted to align the tyrosine to this position of the alignment. Of course, you would try to rather align a glycine there.
So this looks like this is position weight matrix, not an HMM. How do you do with the gaps?
Yeah, so that's 2 slides further down.
Okay.
So the score would then just be calculated. For example, the score for the sequence W, E, I, and so on, A, D, and so on, would just be 6.1, for the W at this position, plus 2.1, plus 2.1 for the U-term, minus 1.2, and so on. And at this position, you would use, for the profiles, sequence profile, you would just use a gap opening penalty, which is always 5.0, minus a gap extension penalty, which is always the same. I mean, in this case, I used 5.0 and 1.5, but it's always the same for the same scoring scheme. And so this sum would be the total score. Now, HMMs, so-called profile HMMs, are actually a very simple extension of this sequence profile. Because the only thing that they add to it is to have this position specific. These scores are position specific. So what they do is they learn the penalties for insertion and deletion based on the statistics of the alignment that you have. So for example, in this case, you have an insertion here, and that means the penalty for inserting something after this column here will be much lower than inserting something after this column, where you have not seen any example. Right, and that makes sense because, for example, you have loops in proteins where you often have seen insertions and deletions in your training alignment, and that means that you want to have less penalty of introducing an insertion or deletion at that position for your new sequences that you find. Okay, so it's just position-specific insertion and deletion penalties for the profilation. So what we do then is comparing these profilation with each other. And BLAST is just a simple sequence search. PsyBLAST, as I already showed you this example, takes the accepted sequences from the first search, builds an alignment out of them, and can use this for scoring with this new profile sequence score that I showed you. Can use this for scoring to get more sensitivity and more accepted sequences, and then put them even in, and you can do in principle 3 or even 5 iterations and get fairly sensitive. So what we did then in 2012 published a paper where we used the same trick. So HEMA is similar to PsyBLAST, but it uses an evolving profile HMM instead of a sequence profile. So it has these position-specific gap penalties. What we did is developed a program called HSplits, which compares a query HMM with a database of query HMM, of the HMMs here. These are pre-calculated, and in order to have these, to get a database of HMMs, we have to cluster the database, of course. So that's one challenge. We have to cluster the database into groups of full-length alignable sequences, build HMMs from each of them, and then we can do this whole thing as an iterative algorithm. And no wonder, because we have more information on the database side, because we are using profile HMMs, we are also more sensitive. What we are quite proud of, though, is that we are also 5 times faster than SciBLAS, even though the calculations that we're doing are much more involved, of course, because we are comparing 2 HMMs with each other, not just a profile with a sequence. So this is, sorry, yes. Yes. It was a mix-up, sorry. So we are measuring the sensitivity in terms of ROC score. So we are taking the database where we know the relationships, called score, and we're counting how many false positives up to a certain threshold we get, as e-value threshold, for example, and how many true positives. We're doing this for all the possible queries in our test data set, and calculating this ROC5 score up to the 5th false positive. But these are details. So this is a way to cleanly benchmark the performance. And what we then get when we compare HSplits with SciBLAS and Hammer is that we actually quite a lot more sensitive. This is the first iteration, but now we can do a second and a third. And you see that also after the second and third, so second and third iteration, we are more sensitive. So what we are showing here is the cumulative distribution of all these ROC5 scores. So for example, this height here gives you the amount of, or the fraction of query sequences that produce an NUC score of higher than 0.5 and or higher than 0.2, for example. Right? So the higher these curves are, the more sensitive this search method. This is one of the standard ways to plot the performance or the sensitivity of search methods. And then after the second iteration, you are even more sensitive, but the difference between these two is more or less stay the same. So HSplits in red is always more sensitive than Hammer or SciBLAS. This is also true for the third iteration, and we've recently included or developed a method that is even more sensitive by clustering, but not only clustering the UniProt database, but by adding local homologs to it. So this is one of the clusters from the UniProt database that we clustered. It has something like 10 sequences or so. We do one iteration of HSplits to enrich it with local alignments. And so we get, instead of just 10 sequences, we get something like 100 or so. So this profile is more diverse, and because it's more diverse, it's better in picking up very remote relationships. Right? So by just adding these sequences, we get quite a significant boost in sensitivity. And I don't have the time to show these results, but the executive summary is basically that there are no disadvantages anymore to HSplits. Maybe for one, except for one, that you have a profile database or a cluster database. So you can't just take a sequence database, you can't just download a sequence database, but you have to either download a specific one from us, which has all these HMMs, or you have to build it yourself. And to build it yourself, we have this software available now called MMseqs that can do very fast clustering. And HSplits is actually the reason why we started to develop these fast clustering and sequence searching algorithms in the first place. So we started this 8 or 10 years ago almost to develop an algorithm that can do very, very fast sequence searching. And Martin has brought this to absolute heights, I would say. And he's going to give a short introduction. How much time do we have still? Because I want to leave enough time for him.
We started 15 minutes ago, so we had
14 minutes, right? 14 minutes. And I take 10 minutes to introduce MMseqs. So this is the same algorithm that we use to cluster the UniProt, but it has evolved so much that now it's actually a very powerful sequence search algorithm. In contrast to HSplits, it's much faster. Why is it so? Later than when we saw how powerful it had become, we realized that it's actually not only useful for clustering but also for sequence searching. And for where do you need to be very fast? Well, you need to be very fast in metagenomics and applications that use NGS data. So, I mean, this plot is, I think, very well known. It shows you the drop in sequencing costs due to the technological developments in sequencing, mainly driven by Illumina in the last years, but now all these long read sequences come, Oxford Nanopore, and so on. So I think this is going to pick up space again. For some time we've been fairly flat here, but I think this is going to become more faster again. So this enormous drop, so this is Moore's law. So this is how CPU performance has dropped. Look, this is an exponential. So this is an enormous pace. This is a factor of 2 every 2 years. Here's the sequencing cost. So it's really amazing how much we've been able to look how much the costs have come down. And of course, that means that now you can generate enormous amounts of sequences that you somehow want to analyze. And for example, HiSeq during the last 3 years or so could sequence, I'm not sure whether this is still up to date, but in 2 days for €2,000, human genome at least 30 times coverage. I guess this is 2 years old, so I don't know where we are now in the story. Last knows. But it hasn't come down much, I think, in the last 2 years. There's an associated boom with the lowered cost of next generation sequencing, and this boom is in metagenomics. So metagenomics is all over the place now. I've put a PubMed alert that sends me all the metagenomics articles mentioning metagenomics every week. It gets longer and longer, and many of them are actually now in Nature, Cell and Science. And one of the reasons why this is so popular now is because people are realizing more and more how important the gut microbiome is for humans. So there's a very intimate interaction between the gut and the immune system and even the brain. And Carsten mentioned the so-called metabolic syndrome that most obese patients have, where you have this inflammation reaction from the visceral fat tissue and so on. So this is very, very strongly influenced by the gut microbes, and that's one of the reasons why metagenomics is experiencing a boom, but it's also because microbiologists are able to study their favorite pets from a completely new angle. Because up to metagenomics, for those, just in case you don't know, means that you don't have to cultivate your bacteria or your whatever microbes you're looking at, fungi or whatever. You don't have to cultivate them because you can do that only for maybe 1–2% of all the known species. But you just sequence them out of their natural environment. You take everything you find in the soil, in the human gut, on the skin, whatever, and you just sequence everything there is. And the sequences are so sensitive that with one single sample, you often find 100 or even more species. Maybe not the complete genome, but a lot of the sequences from those. And you can do that both for DNA and for RNA. And so that means that microbiologists can now study bacteria in their natural habitat in a way that is unprecedented. They can look at interactions, they can look at what biochemical pathways are implemented, maybe not in a single organism, but between organisms, because they exchange metabolites and so on and so on. This is known, for example, in the gut. So this is a whole new area of research that is very, very active now and burgeoning. Also the microbiology and ecology is flourishing. One example for applications is to discover new genes. So we're, as I mentioned, involved in the SocBiN project, where we try to find, we're sampling in extreme environments. So for example, on Iceland, geysers at near to 100 degrees Celsius, and then the Norwegians that are also part of this collaboration have a robot that can dive down to 6,000 meters and sample near hot smokers and so on. And then the interest is to find genes that are stable at quite high temperatures, so they work very well in high temperature reactions in industry, in biotechnology, and that can do some interesting reactions, ligations, whatever, mostly nucleic acid based. There are other people that scour the metagenomics data for new antifungal and antibiotic compounds and for their biosynthesis pathways. And I think this is going to give us really completely new compounds because it gives us a new window into finding compounds that have been developed mostly by soil bacteria, but others as well, to compete against their peers. And so they can kill their peers sometimes extremely efficiently, and most of the antibiotics that we have are from soil bacteria nowadays. So, and I mentioned the human gut microbiome. So that's why a lot of people are now interested in metagenomic sequences, but the problem is that most of those sequences that are sequenced nowadays, there are so many of them that they can't do the function prediction very well anymore for all of them. The tools that they can use need to be very fast, and the fast tools are not sensitive. So BLAST is, you know, a tool that people would like to use, but if you just used BLAST to work on a 6-frame translation of a measured genomic sample from a single, I guess this is one week of a HiSeq 2000 or so, it would already take 1,000 CPU years just to do the BLAST against the UniProt. So what people do is they don't do this anymore. They either use BLAST but against a small database, a representative database, but then they're missing a lot of things that they are not expecting, or they're using much slower tools and use the UniProt. And in any case, they try to gain something like a factor of 100 or 1,000. So our goal was to develop a tool that can have the sensitivity of BLAST, but they can do this in a factor of 1,000 less. So and this is what we've actually finally achieved, and this is what I wanted to tell you about, and Martin is going to give you a short demonstration, or actually a 20-30 minutes demonstration of MMseqs. So as I mentioned, HBLIS is very sensitive, MMseqs is very fast. So HBLIS is much more sensitive than BLAST. MMseqs is about the same sensitivity as BLAST or PsyBLAST, so it can even do profile searches. So whereas in HBLIS we compare profile HMMs to profile HMMs, in MMseqs we compare, just like in PsyBLAST or BLAST, sequences to sequences or profiles to sequences. So how does this work? Well, you need a very fast pre-filter, and you need an alignment algorithm, and this is the key. How to do this pre-filtering, both very fast and very sensitive. And skip this. And the main idea, or one of the main ideas is, well, most of these algorithms work with k-mers. The problem is, if you make the k-mers long, that's good because you make them more specific, and you have to count fewer random hits. Because I think it's good or very useful to realize that if you compare one or many sequences against a big database, so let's say all the 6-frame translated reads from your HiSeq device through all the UniProt, then every pair of matches that you get for a k-mer will cost you a certain amount of time. You can't get that to 0. It will cost you a certain amount of time. That means that the total number of k-mer matches, of identical k-mers, will determine how much time your algorithm will use. So you want to make these k-mers as long as possible, to have as few as possible random hits between sequences that are not related. Okay? Of course, most sequences are not related, where you take a huge amount of sequence and compare it with another huge amount of sequence. So you want to make them long. But if you make them long, of course, you also lose sensitivity, because those long k-mers will not match even among homologous sequences anymore. Okay, so you have a dilemma what to do. And all algorithms that I know of, except BLAST and algorithms that are very similar to BLAST and PsyBLAST, use identical k-mers. For some reason, well, PsyBLAST is doing a really nice trick. They generate similar k-mers. So for every they use 3-mer. For every 3-mer, they generate something like 10 or so similar 3-mers, and so their pre-filter demands a similar 3-mer and another one at maximum distance 40 residues that is also similar to the query k-mer. Okay, so this similarity idea is very powerful, and for some reason has, I would say, been forgotten. And we have used that in order to make these k-mers not just 3 long, but actually 7 long. Why 7? Because we want to make them as long as possible and still be efficient in the computation. And 7 is a natural limit, because if we use more than 7, our index tables get too big for the memory. Okay, so 7 is the natural limit given by the hardware. But of course now, if we have 7-mers, we have to generate really long lists of similar ones, because if you still want to be very sensitive, then you just need not only once to consider one 7-mer, but actually 10,000 or more. So what we do is we take every 7-mer that is in the query protein that we're currently processing, generate up to 10,000 or 20,000 similar ones, and look for database sequences that have one of those 20,000 similar ones. It's still very, very specific, because there are so many 7-mers, right? That 20,000 is still a very small fraction of all of the possible 7-mers. So when you look at this in a more realistic example, these are 2 homologous sequences where we have indicated all the 3-mer exact matches. And you see, this is the true alignment, but a lot of the 3-mers match by chance. But if you now use similar 7ers, you have roughly the same number of matches here, but you have no false positive matches anymore. That's because a 7er is much more specific than a 3mer. And through the similarity, we get still a sufficient number of hits. So this is, I would say, maybe the most important idea in MMseqs. And another way to show this is that with 3mers, you roughly get 1/20 chance of a random hit, right? 1/20 to the power 3. And if you have homologous sequences, it means, so the 25% sequence identity, the probability to have 2 amino acids match in the region of the alignment where you have 25% sequence identity is 0.25 to the power 3, so it's 1/64. Now if you take an exact 5mer match, you go down from 1.2 times 10 to the -3 to 3 times 10 to the -7. And with the probability of a match for homologous sequences, you go down to 1/1000. Now this is too low, as I said, but now you take the similar ones, you go to 6mers, but you look at a list of 20 similar ones, then you are back actually at 3 times 10 to the -7 for the probability of a chance match, but you're at 1/50 for the probability of a match for homologs. Okay, so that's the trick. And then I think I have no time to talk about this anymore, how the actual scoring works. But I wanted to tell you that about the improvement that probably gave us the most increase in the last year or so that we've introduced among many other changes. So we reasoned that this algorithm is nice that we have, so we can score all these similar k-mers. They are very specific, so that's nice. But we're still accumulating noise over those hatched regions here, whereas our signal comes only from a linear region. So our signal grows proportional to the length of the alignment, but the noise grows quadratically. That's not very nice. So we thought, how can we make this more sensitive, more specific? And the idea is actually, first we thought we have to score diagonals. So each diagonal is one of those lines, and we can just, instead of scoring sequences, we score sequence and diagonal together. And then we just look at which of the diagonals in which sequence have accumulated a good score. In principle, this would work, but the problem is that you need to store the scores of not only 10 to the 8 sequences that you're searching against in your database that you're searching, but also times the number of diagonals. And when you compare a sequence of length 300 with a sequence of length 300, you need to store 600 diagonals here, each one with a number. This one would be 0, -1, -2, and so on, +1, +2, and so on. And just to hold this in memory is completely impossible. So this doesn't work, but that brought us to an idea that combines both advantages. The idea is we don't store for every sequence in every diagonal the scores, but we just store for every sequence the diagonal that we've seen as the last match. And we go through the query sequence from left to right. So let's say this is the query sequence. We look for the query sequence in all the database sequences, whether we find a similar k-mer. And let's say the first one we find is this one. The second one we find is somewhere up here. That's actually covered by this legend. The third that we find, when we find it, we see that the previous diagonal was a different one because it was down here and the other one is up here. Then we find the third, and then we find the fourth, and the fourth actually has the same diagonal as the third. And whenever we see that the previous hit had the same diagonal, we have a match, because that means we have 2 in one diagonal, and that occurs very rarely by chance. And so what you see in green, this one is actually false, it should be blue, but what you see in green is all those that give a score. This one, for example, this blue one, doesn't give a score, because it was not the previous one that had the same diagonal, but it was the one three before, so it does not trigger the scoring element. All of those green ones do, because this green one, for example, came directly after this blue one and it had the same diagonal. So this makes the algorithm both as fast as the previous one, but much more sensitive. And with this algorithm and other tricks to be local in the cache of the computer, so we don't have to access the random memory and so on, we get sensitivity. So this is again an AUC plot where you want to be as high as possible. We get sensitivities that are very close to blast and swipe, but at 30 to 50 times of the speed. If we make our program very sensitive, so this is the dark blue line. If we make it faster but less sensitive, so this is this light blue line, we're even 380 times faster or 2,000 times faster for this version. And you see there's nothing that even comes near to the, for example, the 380 times faster version from the other programs. So for example, Diamond or Last that I used a lot in metagenomics. They're actually slower and less sensitive. And as I mentioned, we can also do profile searches. And here, this is the first iteration of BLAST, and now we can compare it to the second iteration of PsyBLAST, which is the light gray line, and the third iteration of PsyBLAST, and the fourth iteration, and compare this to MMseqs, first, second, third iteration. You see that we are actually quite a bit more sensitive, and we're not even more sensitive. We also have much lower false discovery rates, which are a problem for iterative searches, but I don't have enough time to tell you about this. And I think, Martin, you are going to give us an example, right? The clustering, I think I can't mention anymore.
So how do you choose the subset of the k-mare? So if you have the 7 k-mare, you say you take some deviation from it.
It's just the score. We calculate the blossom score. So we go through this query for every 7 overlapping. For every 7 we generate this list of similar ones.
What
and the list of similar is simply those that score above a certain blossom score with the original one. Okay. So it's just using the blossom matrix. And you need a very fast algorithm that actually can generate this list in O of in the complexity of the length of the list. So that is important.
Okay, so I'm gonna introduce you to MMseqs. This should be a live demo, so I will show you how to use MMseqs, and the idea is to give you some ideas how to use it, how to install it, and initially how to compare MMseqs, so in BLAST. So I will do some live searches, and then we can see the speed up and how well it works. And then I will give some general usage example, and afterwards I will explain a bit about applications. So what are we going to do with the software, and then also explain you how you might be able to do the same analysis that we are going to do with the software. So to do this, I have installed on my machine, so my machine has 4 cores. I have installed BLAST, and I will gonna search a query set with around 6,300 queries against the database with around 1.6 million sequences. Just one question, is everybody familiar with this kind of interface, like console? Did you guys use that? Everybody?
Yes.
Perfect, good. I was a bit scared. Okay, so what you're gonna do now, we start to run BLAST. With this query set, we run it on 2 cores. This is minus num threads 2 parameter, and we have some kind of e-value threshold. And so this will run for a bit now. So while BLAST is running, I'm gonna install MMseqs. So what you will get when you have MMseqs is you will get a tar which has some kind of static compiled MMseqs, or you can have the source code. I will just do the simpler way now. So I will extract this tar that you will get. And this tar has some files, it has a print folder, there's a data folder. The data folder contains some blossom matrices, and it has some shell scripts. But yeah, this is not really so important. So you just unzip it somewhere where you want to have it. What you have to do then is to set the n variable called mmdir, which should show to the directory where you install MMseqs, so I can use this variable later to detect the blossom matrices and the shell scripts. And what we have to do then again is we have to get the path. So we set the path to the bin directory, and now I have to do a hack. This hack I have to do because I have a Mac, and the static compiled version is for a Linux version, so I just compiled for my local compiled MMseqs to the bin folder, and that's how it is. And then you can run MMseqs by just calling MMseqs. And when you run MMseqs, it will show you all these different modules and things that are implemented in it. And so this is all the overview of all these like tools we developed and the workflows we created to search sequence data. So let's see how BLAST is doing. Okay, BLAST is still running. So now we're gonna go and do the same thing with MMseqs. Let's go for this. So what we have to do is we have to create first a database. You have to do in general do the same thing with BLAST. So if you want to use BLAST, you call this tool make database, and this will take your FASTA file and convert it into a database. So MMseqs has something similar. You call this MMseqs binary and just say create database, give it your FASTA file and then some name where you want to output the database, and then it will run. And it will now start to convert it to the internal MMseqs format. So MMseqs will always print you out, for example, a version which is running. So this is the GitHub version actually of MMseqs, which is useful if you give some bug reports or something. You can just say, oh, I have this version. There's some problems. Can you please fix it? That helps us a lot. Okay, now we have the database. What we can do now, we create an index from this database. So what does this do is it goes over all the sequences, it counts the k-mers, how often they occur, and then start to fill up the index. So this is around 1 million sequences, and each dot that you see here is 10,000 sequences. So creating the index takes some time. This doesn't run really parallel, it runs on one CPU. So if you want to, for example, create an index of the UniProt database with around 50 million sequences, you need like 1 hour or so to create the database. But you just have to do this once. You pre-compute this index once, and then you can reuse it all the time whenever you want to search against the database. So let's stop it at this point and see, okay, BLAST is still running. What we do now is we just make the same search as BLAST does. What we do for this is we call MMseqs, and then we call the search program, and we give it the query database, which contains the 6,000 sequences, and we search it against the database where already pre-computed the index, and some other parameters is threads too, so it also runs on 2 CPUs. Since I have 4 cores on this machine, 2 is for BLAST and 2 is for MMseqs now. And I just gave it the -s parameter, which is the sensitivity you can adjust with this. So -s1 is the lowest sensitivity of MMseqs. So it's not exactly a one-to-one comparison with BLAST, but just see how fast this is. So MMseqs really likes to talk a lot. So it tells you everything it does, step by step. It tells you all the parameters that is chosen, which is also really useful. So if you once you analyze some data, MMseqs will exactly show you what are the parameters and that are used. So it's really nice for reproducing afterwards the same result. At the moment, it just reads in the index. It also tells you how much memory it needs here. So it needs around 6 gigabytes to run at the moment for this data set, out of my roughly 60 gigabytes. And then it tells you, okay, it's already done with the pre-filter. Oh, it's okay, it finished already running actually. So the pre-filtering score calculation, what Johannes was telling, there's the part that has to be really fast. Took like 3 seconds for this 6,000 sequences against this 1,600,000 sequences. And after the pre-filter, we call the alignment module, and this is the alignment module output. And to calculate the alignment for all of these took like 26 seconds, and BLAST is still running. So I let BLAST run for like 2 hours once on my machine. In the meantime, I went to a lottery and came back, and I thought, oh, BLAST might be finished, but no, BLAST was not done. And so I estimated the time that BLAST would need for this kind of analysis, and it would be 15 hours. So it runs around 15 hours, while MMseqs is now roughly around 30 seconds, something like this. On my local machine. So with this, we more or less can see MMseqs can be really fast. And you have also the advantage of being able to adjust the sensitivity based on your use case. So, for example, you want something really fast and you just go with a really low sensitivity, you can then go up to the sensitivity of blast. So, what is the output that you get here? So, MMseqs uses an internal format which might be not really handy for most people to use probably. It shows you the alignment in a tab format, and the first column is the idea that MMseqs actually is giving the sequence, and it tells you the e-value of a hit and the start and end position of the query and the start and end position of the target and the score it produced. So if you really want to use the results in a more handy way, you can format the alignment in different formats. So at the moment we support the BLAST format because it's really useful to have, because the most tools that go downstream then uses the same format. So I just formatted the alignment to the BLAST format. You can do this with the call MMseqs format alignment. And with this you then see, this is more easy to read. So you see on the left column, this is the query. On the right, then the second column is the target that got hit. You can see the sequence identity, and you get all the information that you more or less need for your alignment. Okay. MMseqs is also able to cluster databases. Let's check the time. Okay. To cluster databases. So we created a clustering module in MMseqs, which was actually the initial plan for MMseqs. We developed MMseqs to be able to be really fast in clustering huge databases down, because we need this for the HH Blitz databases for our MSAs. And let's change that. Just to see that, it's the same way how you call it. So you call MMseqs, then you say clustering workflow. You give it the database you want to cluster. At this point, I give it the SwissProt, which has around 500,000 sequences. And then I say what should be the result file, and I have to do the temp file. And I can then start to give it parameters like min, sequence ID, or the coverage I want, the criteria on which I want to cluster things together. So MMseqs start to cluster, and there's a lot happening. MMseqs likes to talk. We have this clustering workflow is an iterative workflow, so it clusters down in steps. So it goes down from a really low sensitivity to high sensitivity, because what happens if you start clustering 500,000 and then you go down to 300,000, and then you take the representative sequences of the clusters of this 300,000 clusters, and then you go with a higher sensitivity down to maybe 50% sequence identity, then 40, and then 30. And this saves you a lot of CPU time, and yeah, it makes this workflow really fast, and also really creates really nice clustering results. So now it's pre-filtering, so the pre-filtering runs with 400,000 sequences against 400,000 sequences. Since I did not compute, okay, I did not create the index here for this file. So if you don't create the index, which I did before, this like create index, which takes some time. If you don't do it, MMseqs will start creating the index for the pre-filter. So it does this at the moment, and now it runs. And you can see it's actually really fast. It's already at around 200,000 sequences that are done. So 300,000 sequences or so, and the pre-filtering is done. So with this the all against all comparison, the biggest bottleneck that we have at the moment is actually still doing the alignment module. So we were able to bring down the pre-filtering. The pre-filtering is now so fast that the main bottleneck is now starting to really compute the alignments for all the hits that the pre-filter found. So this might take some time. So let me just stop this at this point. I just want to show you what's actually the output that you get out of the clustering. And you can see it. So I have already pre-computed this clustering. It takes around, on this machine, 3 minutes or 4 minutes to cluster down this 500,000 sequences. And now we create a useful format. And then it will print you out TSV file where in the first column you see the cluster representative, and in the second column you see all the members that are in this sequence. So each line is one connection. So let's go to a cluster this big. So let's have a look here. So the Q6GZU6 would be the representative, and it contains itself, which makes sense. And then it contains, oh, it does not, sorry, that's not a good example, sorry. Oh, here, that's a good example. The P0C6F9 contains itself, and also contains 4 other sequences. So yeah, that might be, so you can create different formats of your clustering how you need it. You can include the sequences or not, and you can just extract the representative sequences. So clustering is actually really handy to use also. So okay, let's show how we do this. So we were interested to use MMseqs for a metagenomic project. Did anybody hear of this Tara Ocean project before? Okay, not so many, that's good. So the Tara Ocean project was started several years ago, and there's one boat, it's called the Tara. Let's go to the boat first. This is the Tara. And the Tara is supposed to sail around the world and take samples from the ocean on different points. You can see here, for example, the Tara is sampling in different depths, like 10 meter deep, 100 meter deep, 1,000 meter deep, and it goes through the ocean and samples at various spots. And here you can see the map until the year 2013, where they were sailing around. So you can see they have a huge coverage already. So they have a current sailing trip around the Pacific Ocean, around Japan, in the areas around the Pacific. So they are constantly still collecting a lot of data, and they want to map the diversity of the ocean. They're interested, so what is there, what we might not know. And there was a paper published last year, which is a gene catalog of this Tara Ocean data. And what they did, they tried to create reference gene catalog. So they want to know what is the diversity of this of the ocean. So what they did is they used seven terabases of these samples and created with MoCut 111 million coding genes, and then added to this 100 million genes also genes that got predicted by other expeditions through the ocean, like the GOS or the MMI, and some viruses that are detected. And then they clustered this whole catalog down to remove all the redundancy, and they ended up with around 40 million non-redundant sequences that they could find in the ocean. And there's a lot of novel sequences. So what you can see here is, I told you they sampled at different depths in the ocean. And on the right side you see more or less a bar chart of what is known and unknown. And you can see as deeper you go in the ocean, as there's more unknown that we don't know, that we cannot match to anything in the database. So there's a high chance for us maybe to make some impact with our remote searches that Johanne was introducing, to maybe make sense out of the sequences that we don't know what it is. And so in the paper, they started to make a function annotation using BLAST, and they ran this 40 million sequences against the ECNOC database, which is an ortholog group database, to map functions. And to run this against this ortholog database, they needed around 170,000 CPU hours with BLAST. So running this 40 million against this 6 million. And I did the same thing with MMseqs in around 180 times the speed of this. And I want to show you how I did it. So, it shouldn't start. So to run this example, what you can do, so we want to reach the same sensitivity as BLAST, so we want to annotate as much as BLAST, but we want to be as fast as possible. So what you can do with MMseqs is to tell MMseqs, start with a really low sensitivity and search this 40 million against this 6 million sequences, and then take every sequence out that you could annotate already, and then go deeper in sensitivity, so make it more sensitive, so you can step by step go deeper in sensitivity. So the first fast searches will be done really fast. So you will be annotating around 20 to 25 million sequences with MMseqs really fast, and then for the rest you need a lot of time to spend for making it more sensitive. And to tell MMseqs to do this is fairly simple. You just tell MMseqs it should start with the sensitivity 3 and then go to sensitivity S7. S7 is more or less the sensitivity that BLAST has. And then, yeah, it goes through the catalog, and you will get one result file at the end. So you don't have to care about the details. And let's run this. So it makes the same search as before, more or less. You can see it needs some seconds to do the pre-filtering with the 6,000 sequences. So what you also can see here, actually from the output, maybe quite interesting, MMseqs now produces around 112 k-mers per position. So per residue position in the query, we roughly create this amount of k-mers, which we start then to match in the index. And yeah, now we compute the alignment. What else is interesting to see here? Yeah, you can then see in average, you can get some statistics, how much database matches you have, how much double diagonal matches you have. Johannes was talking about that we want to have two hits on one diagonal until we accept this as a hit. Okay, now the alignment is done, and now it starts the next pre-filter, and the next pre-filter is more sensitive now. It now creates around 850 k-mers per position. We see we have a huge amount of database matches per sequence, and also the double diagonal hits go up. But we can do this really fast because there's just 4 sequences left in the set of the 6,000 sequences, because we could already annotate nearly everything in the first iteration. So we are done and annotated all the sequences for this example. So this is really handy for annotating a huge amount of metagenomics data. Okay. And I have one more, a bit more complicated example, where they try to make a taxonomical annotation for this whole catalog. So they want to have, for all these 40 million sequences, they want to annotate them and know to which taxa it belongs. Here on the right you can see there's the composition of the gene catalog, how they did it. And they used a bit of a difficult protocol for doing this. And it's called dual BLAST-based last common ancestor. And the protocol works the way that you search this 40 million sequences against, for example, a uniprot. They use a uniprot at this point. Then whenever you find a hit with an e-value better 10 to the minus 5, you extract the alignment. So you extract the target alignment sequence, so you have the start and end position, so you take this part out. And then you search with this part against the same database again. And then you extract every hit that has a better e-value than the e-value you could find before of this fragment. And then you start to compute the LCA. So this is already a quite complex flow. And I can show you that with MMseqs. You can do this quite simple by first doing the same thing. So the first step is we start to search with this queries against the database. Just run this program and explain the next step. And then you can call one util that we created in MMseqs to filter databases. So we filter the database then, so the result that we receive from this, and extract always just the top hit, so we get the best hit out of this. So we have a new database called result top hit, and then we have a command which can extract the aligned region. So we are able to extract this domain we are interested in. Let's see if we're done, not yet. And after we create a new database which has this high scoring regions, and then we search this high scoring regions against the database again, and then we start a bit of magic. Let's see. Okay, it's called it. Now we will filter the database, so now we have all these top hits. We can have a look at how many lines we have here in Linux. So, okay, we could find for everything, we could find a top hit. So let's extract the regions. So now we create a new database with this high scoring regions, and now we start searching them against this 1 million sequences again. And then we just format the results with the top hit into this M8 format, this blast tab format, and we then format the hits that we find from the eight HSPs to the M8 format, and then we can do some tricks with org to just get all the hits that have a better e-value than the other one by joining these two result files together. So this will now print us out all the hits that have that fulfill the criteria of e-value better than V1 and it's this high scoring region. So it's actually simple, just with 5 commands you can create this workflow. And yeah, so summary, we have a blast-like search, which is really fast. We can cope with a huge amount of data. We can run it on several machines. We can cluster it. We have profile searches. And yeah, we are going to work in future probably on a nucleotide version of MMseqs, so to also being able to assemble data with MMseqs. And yeah, I want to thank all the people that are involved in the development of MMseqs. And it's not just me. It's actually Maria who started the thing. Then a lot of people like Milo, Clovis, and Lars, which develop tools. A lot of this like utils that helps a lot to use MMseqs. And also our beta testers, we have people from the EMBL who are starting to use MMseqs to cluster the ECNOC database, or people who are interested at the UniProt to cluster the UniProt, and people at the Max Planck who support the toolkit of HHPrad and use MMseqs now for clustering the database. Okay, thanks a lot.
Now it's time for questions.
Yeah, it's not yet. We are not yet really coding it, but we have ideas what we want to achieve with it. So I think it would be, especially for the long read technology, it's really useful to use this kind of approach for finding, for especially assembling genomes. But yeah, we're not yet there.
I'd like to use nucleotide version, because I don't know.
Okay. A lot of people ask for the nucleotide version actually already for clustering.
Did they have sequencing machine on board on Tara?
Oh,
you know this?
Actually, I think they don't. But they have a dry lab, and I think they are freezing the samples. I heard a talk about how they do it. So they freeze them down, and they have like certain protocols to assure that the quality is good. But they don't have a sequencer.
Any more questions? No? Thank you.
I think now we will have